Hello, I'm Caroline Clark, conference producer at Technology Networks, and I'm here to introduce the final talk of the day. In vivo CRISPR activation platform enables modeling of aggressive lymphoma. I'm pleased to welcome Yexuan Deng as your presenter. Yexuan is a postdoctoral fellow in the Herald Laboratory Blood Cells and Blood Cancer Division at WEHI. Her work utilizes multiple CRISPR-Cas9 genome editing systems to achieve depletion of activation of specific genes in eukaryotic cells, which has been published in Immunity Nature Communications. Yexuan characterized the CRISPR activation mouse model, which showed high efficiency in activating gene expression in hematopoietic system. She used this system to develop a mouse model of double hit lymphoma by inducing pro-survival protein BCL2 expression in UMIC transgenic DCAS9A SAM knock-in hematopoietic stem cell and progenitor cells. A warm welcome to you, Yexuan. Following the presentation, we will have a Q&A session and we will welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of your player. You can also use this feature to connect with attendees and discuss the talk, so feel free to take advantage of the chat system by introducing yourself and sharing your thoughts during Yexuan's presentation. If you experience any technical difficulties during the presentation, please request help using the technical help icon on the menu on the left-hand side of the player. Now, without further ado, I'll hand over to Yexuan to begin their presentation.
Hey, and thanks for the introduction, Caroline. Hello, everyone. Thanks for coming to this symposium, and thanks to Technology Networks for your invitation. Today, I'm going to talk about in vivo CRISPR activation platform enables modeling of aggressive lymphoma. I'm sorry. Lymphoma is a type of cancer arising in cells of the lymphatic system. There are two main kinds of lymphoma, Hodgkin lymphoma and non-Hodgkin lymphoma. Hodgkin lymphoma can often be cured by chemotherapy. However, non-Hodgkin lymphoma prognosis varies by subtype. Diffuse large B-cell lymphoma is the most common subtype of non-Hodgkin lymphoma. Some cases have high-grade morphology and frequently harbor MYC and BCL2 and/or BCL6 translocations. Double-hit lymphoma was previously classified as DLBCL with rearrangements of MYC and BCL2 or MYC and BCL6. Now it is recognized as high-grade B-cell lymphoma. It has poor prognosis when treated with conventional therapies such as standard R-CHOP chemotherapy. Mimicking MYC and BCL2 aberrations is helpful for developing therapeutic approaches of DHL treatment. The c-MYC transcriptional factor regulates about 10,000 genes, including many that control cell growth and division. Deregulated of c-MYC expression is detected in up to 70% of human cancers, including lymphomas and leukemias. Early in 1985, Jerry Adams has developed a mouse model which employs a transgene, mimicking the translocation of c-MYC and IgH enhancer elements characteristic of human Burkitt lymphoma. Immunic mice develop B-cell leukemia lymphoma-like malignancy when 100% penetrance. It's shown that in these immunic lymphomas, they normally express high levels of pro-survival protein MCL1, which is essential to sustain c-MYC-driven lymphoma cell growth. However, another pro-survival protein BCL2 expression was shown at lower levels in these lymphomas. So what are the pro-survival protein? Intrinsic apoptosis triggered by cellular stress or developmental signal is regulated by the balance between pro-survival and pro-apoptotic BCL2 protein family members. Under certain stress, BH3 only proteins can inhibit pro-survival protein activity or directly activate pro-apoptotic proteins back to induce mitochondrial-mediated apoptosis, while pro-survival proteins can bind to back to prevent their functions and lead to cell survival. Cancer cells can evade apoptosis by overexpressing one or more pro-survival proteins. This can be overcome using BH3 mimetic drugs, such as MCL1 or BCL2 inhibitors. Despite the low BCL2 expression shown in normal immunic lymphoma model, Andres Strasser pointed out the combined overexpression of MYC and BCL2 can accelerate lymphoma development in an immunic BCL2 mouse model. This is like double-hit lymphoma with increased MYC BCL2 levels. However, these tumors were developing so fast and identified as immature hematopoietic progenitor tumors with no Ig or TCR gene rearrangements and did not represent classical B-cell lymphomas. Additionally, their derived cell lines were unable to be cultured in vitro, which limits the application of this model. Therefore, we are seeking for a proper mouse model that can mimic double-hit lymphoma and can be used for drug discovery. CRISPR editing system is an elegant tool to achieve gene editing both in vitro and in vivo. The wild-type Cas9 with its designed sgRNA sequences can deplete targeted gene expression in cells. In 2015, Feng Zhang Group reported a SAM system, which is using a catalytically deficient Cas9 variant linked to a transcriptional activator VP64. This complex can be guided by sgRNA to the promoter region of the targeted gene and activate gene expression. It also has a designed MS2 stem loop in the sgRNA scaffold that can be recognized by MS2 and lead to the recruitment of two additional transcriptional activators, P65 and HSF1, to increase its activity. This system is proven as more efficient in activating targeted gene expression than some other CRISPR activation screens. CRISPR activation systems. For ease of manipulating cells, we integrated the SAM system originally described in a two-vector configuration into one single lentiviral construct. We linked the dCas9 VP64 while T2A sequence to the MS2 P65 HSF1 expression cassette. In addition, we incorporated an EGFP sequence as a selection marker while a second T2A sequence downstream of the dCas9 VP64 T2A MS2 P65 HSF1 coding sequence. We can see from this little animation, the dCas9 VP64 complex is guided by the sgRNA to the targeted gene, the promoter region, and starts to achieve gene expression. Meanwhile, the MS2 P65 HSF1 complex is binding to the sgRNA scaffold and acts as more transcriptional activation signals to this promoter. We can use EGFP as a selection marker to sort for cells containing this CRISPR-A system. To validate the efficiency of this CRISPR-A system in vitro, we introduced the lentiviral vectors encoding the CRISPR-A cassette and sgRNA targeting the BCL2 promoter into cell lines derived from immunic mice that developed lymphoma. We could detect increased BCL2 protein levels by Western blotting in BCL2 sgRNA transducer cell lines. Since the sgRNA vector has puromycin resistance element, BCL2 protein levels were even getting higher when cells were selected by puromycin. We have known that immunic lymphomas are highly reliant on the pro-survival protein MCL1 for their survival, and thus are sensitive to the MCL1 inhibitor, S638 845. The elevated BCL2 expression in these immunic lymphoma cell lines can rescue the loss of MCL1 and increase the cell resistance to the MCL1 inhibitors. That means our CRISPR-A system can successfully induce functional BCL2 protein expression in cell lines. To expand the application of this system, we generated a CRISPR-A transgenic mouse model based on our one-vector system. We targeted the CRISPR-A coding cassette into mouse ROSA 26 locus and used the CTV vector, which has a LOXP flanked stop cassette between the CGA promoter and the SGN and the cDNA. We then crossed the transgenic mice with pre-deleted mice to remove the stop cassette, allowing constitutive expression of the CRISPR-A components in all tissues. We refer to the resulting string as the DCAS9A SAM knocking mouse. To evaluate whether our knocking mice show normal development, we first confirmed that DCAS9A mice showed similar in aging compared to wild-type mice, which indicated these mice have no defects in lifespan. Then we checked impacts of DCAS9A cassette expression on cellular composition. We analyzed the hematopoietic compartment because it is particularly sensitive to cytotoxic stress. We checked T cell distribution in the thymus, B cell lineage, and myeloid cell distribution in the bone marrow. It turned out there is no difference of these two tissues between DCAS9A mice and wild-type mice. We also checked T cells, B cells, and myeloid cells distribution in the spleen, T cell and B cell distributions in the lymph node. It turned out there is no difference of these two tissues between DCAS9A mice and wild-type mice. Since we have confirmed that our CRISPR-A knocking mouse shows no signs of disease and doesn't cause any cytotoxicity to the hematopoietic system, we sought to use it combined with the immunic mouse model to develop an aggressive lymphoma model. We chose double-hit lymphoma, which didn't have a suitable mouse model to mimic the disease. We first crossed female DCAS9A knocking mice with male immunic transgenic mice. Then we performed the hematopoietic reconstitution experiment. We harvested fatty livers from E14.5 embryos of the DCAS9A heterozygous immunic transgenic genotype. The E14.5 fatty liver is a rich source of hematopoietic system as hematopoietic stem and progenitor cells. Then we infected fatty liver cells with lentiviruses that express BCL2 sgRNA constructs in vitro. Next, we injected these cells through IV injection into lethally irradiated recipient mice and waited for tumorigenesis. According to the survival curve, we found that mouse transplanted with HSPCs expressing the BCL2 sgRNAs developed lymphoma more rapidly with median latency of 6-8 days compared with median latency of 102 days for control mice, which were transplanted with HSPCs expressing non-targeting control sgRNAs. To characterize our mouse model and compare with features of human double-hit lymphoma, we performed Western blotting and verified the high expression of BCL2 in immunic DCAS9A SG-BCL2 lymphomas. These lymphomas also reviewed a B-cell phenotype as CD19 and B220 double positive that is observed in human DHL. Analysis of the immunic DCAS9A SG-BCL2 lymphomas and cell lines reviewed a monoclonal origin of malignant cells. To further characterize our mouse model, we performed the RNA-seq and compared the transcriptional profile of our developed lymphomas with a murine pre-BALL model. It revealed clear differences in the top 200 variable genes, including the upregulation of specific genes EP300 and stage 6, which is also observed in human DHL. We then used the pathway analysis tool to figure out the upregulation of MYC and DLBCL pathways in our developed lymphomas compared to pre-BALL. This data confirm that we have established a model of aggressive B-cell lymphoma that has characteristics of human double-hit lymphoma, including the tumor development is very aggressive. The lymphoma cells originate from the B-cell lineage. Lymphomas express high levels of BCL2 and c-MYC. RNA-seq analysis revealed pathway aberrations that are characteristics of human DHL. Venetoclax is a type of BH3 mimetics which inhibits BCL2 activity. It's been approved by FDA for the therapy of CLL and AML and is in clinical trials for several other cancers. An important clinical issue of venetoclax is the emergence of resistance in patients undergoing therapy. To identify new therapy resistance factors of venetoclax in human DHL, we sought to use our generated DHL mouse model to perform a CRISPR activation screen. We first generated some immunic lymphoma cell lines derived from immunic DCAS9A SGA BCL2 lymphomas and validated high BCL2 levels in these DHL-like cell lines. Then we used venetoclax to treat these cell lines. The non-targeting control cell lines showed similar features as the wild-type immunic cell lines, which are mainly relied on MCL1, thus are resistant to venetoclax. However, unlike control cell lines, DHL-like cell lines showed their high sensitivity to venetoclax at the average IC50 of 0.11 micromolar. Next, we started our CRISPR activation screen in two of the most sensitive cell lines, 214 and 216. For each cell line, we transduced it with the whole genome CRISPR-A sgRNA library Caprino of 6 replicates and expanded cells for 2 weeks to permit induction of gene expression. We divided each replicate into 3 parts and treated cells with vehicle or drugs. One was treated with DMSO as a control. One was treated with venetoclax and IC50 dose, and later on at the IC50 or IC100 dose. The last one was treated with venetoclax at the IC80 dose, after culturing for 2 weeks for the drug selection and cell recovery. We collected cell samples for the next generation sequencing. NGS data showed that at all concentrations, venetoclax treatment is lead to a strong enrichment of a subset of sgRNAs compared to the DMSO-treated control samples. We found enrichment of sgRNAs upregulating 2 pro-survival proteins, BCL-XL and MCL1, which are expected to mediate resistance to venetoclax. This indicates the reliability of our screen. Interestingly, we found sgRNAs targeting promoters of different BCL2A1 variants shown in both cell lines and both doses. BCL2A1 gene encodes another pro-survival protein A1, also known as BFL1 in human. A1BFL1 is shown to be upregulated in different cancers, including virus, blood cancers, and solid cancers. Some publications also described its role in inducing chemotherapy resistance and BH3 mimetic drug resistance. So A1BFL1 is our favorite hit from this screen. To confirm that upregulation of A1 confer protection from venetoclax-induced killing, we transduced sgRNAs targeting B-cell to A1 promoter in both DHL cell lines and validate A1 overexpression by Western blotting. Then we performed the cell computation assays in vitro. We generated the parental DHL cell line expressing high EGFP and mixed 1-in-1 with A1 activated DHL cell line. We treated the cell mixture with venetoclax at the IC50 dose. After 6 days, we could see that A1 activated cell proportion has been raised up to 100%. Therefore, we confirmed that A1 activated cells possessed a striking survival advantage over parental cells in the presence of venetoclax. To conclude, we identified the pro-survival BCL2 family protein A1, also called BFL1 in human, as the driver of venetoclax resistance in DHL cell lines. There are some advantages of A1 as the drug target in the clinics. Some papers show that the absence of the A1 protein shows no impacts on embryonic development, tissue development, and lifespan. Thus, using drugs targeting A1BFL1 is expected to cause no on-target toxicity to healthy cells and could likely be used in combination with venetoclax or MCL1 inhibitors. After I explained how to use our CRISPR activation mouse model mimicking double-hit lymphoma, I want to propose more applications based on our CRISPR-A mouse. Since we have confirmed our CRISPR-A system worked well both in cell lines and in vivo, we sought to explain its application in primary cell lines derived from dCAS9A knocking mice. We isolated naive B cells and T cells from mouse brain and transduced these cells with the sgRNAs targeting BCL2 promoter. We could see increased BCL2 levels by flow cytometry and Western blotting. We also transduced another sgRNA that targets CD19 promoter in both B cells and T cells. As expected, it showed increased expression levels of CD19 B cells. Interestingly, CRISPR-A system also activates CD19 expression in T cells, in which CD19 is normally transcriptionally silent. Furthermore, we isolated mouse embryonic fibroblasts from E14 embryos and transduced with BCL2 sgRNAs, where we could see the increased BCL2 levels. We also performed similar experiments in basal cells that isolated from mouse mammary glands with sgRNAs targeting BCL2 promoter or GATA3 promoter. We could detect both gene activation in basal cells. This data indicate that we can achieve gene activation in different primary cell types, and this model can be used for ex vivo experiments. Besides exploring drug resistance factors by CRISPR activation screen in cell lines, our lab is now focusing on utilizing in vivo CRISPR-A screens for different purposes. One of the purposes is to search for novel oncogenes that drive different types of cancers. Again, we are using the immunic mouse model to look for genes that accelerate lymphoma development. We first transduced the dCAS9A immunic fetal liver cells with the whole genome sgRNA called Caprino. This Caprino sgRNA library has two parts. Each part has 3 sgRNAs targeting the same gene promoter. After mice got sick, we harvested tumor tissues and performed the next generation sequencing to recognize enriched sgRNA sequences and identify specific genes that lead to tumorigenesis. We could look from this survival curve. We could see that the significant acceleration of lymphomas in the presence of Caprino library compared to the control mice. We have sequenced these samples and will validate interesting hits in subsequent experiments. Another mouse model we're using is the p53 heterozygous breast cancer model. As I think most of you have known that TP53 is the most frequently mutated gene in invasive breast cancer. TP53 is mutated in 30 to 35% of all cases, while is mutated in approximately 80% of triple-negative tumors. Previous reports showed that one allele loss of p53 in the mammary epithelium was sufficient for spontaneous development of mammary carcinomas in mice at the latency of 8 to 14 months. Therefore, we isolated basal cells from mammary glands of mCRA, a transgenic TP53 flox class and DCAS9 SAM heterozygous donor mice, and transduced them with a specific transcription factor sgRNAs that designed for CRISPR activation. After transplanting basal cells into cleared fat pads of DCAS9A recipient mice, we are waiting for tumor growth and will harvest tumor samples for NGS. The other purpose is to seek for genes that can improve immunotherapy, for example, the CAR T cell therapy. CAR T cell therapy is a promising immunotherapy that depends on cytotoxicity, T lymphocyte activity to kill cancer cells. However, the success of this therapy is limited by the inefficient trafficking of CTLs into solid tumors and the failure of CTLs to persist and maintain their cytotoxic function. To enhance CTL activity in immunotherapy, we are performing CRISPR activation screens to explore CTL intrinsic regulators of these processes. We first isolated and activated CD8 positive T cells from DCAS9A OT1 mice spleen, and then transduced these cells with a transcription factor sgRNA library. We injected the REC1 recipient mice with sorted BFP positive transduced T cells. We can see it's like 30% something in this figure. After 35 days, we collected spleens and lymph nodes from these mice and prepared samples for NGS. We will validate interesting hits from the result and figure out potential targets that can be used for improving CAR T therapy. To summarize my talk, first, we generated a CRISPR activation mouse model that can induce targeted gene expression in vivo. Then we generated an aggressive double-hit lymphoma mouse model using our CRISPR-A platform. We identified A1 BFL1 as a novel drug target to overcome venetoclax resistance in DHL. Then we also developing different strategies to investigate oncogenes in driving lymphomas and breast cancers. We are also seeking for new regulators that can enhance cytotoxicity T cell activity in immunotherapy. This is the acknowledgement. First, I want to thank my supervisor Marco Herold to help me with this paper. And I want to thank Maggie, Gupna, Jerry, Amali, Andrew, Martin, and Lane for all her help to finish this paper. And also Emily is helping with the things of the T-cell therapy and other members from Hero Lab. I also want to give a big thank to Sarah and Gemma, who has helped me quite a lot for this paper, especially Sarah was doing the CRISPR-A screen in cell lines, and also other lab heads and other people from our BC division at Weihai. And I also want to thank my co-supervisor, Jen Weisweder, who is helping me with developing all the breast cancer model screens. And I want to thank Chen Zhao, my PhD supervisor, Chen Zhao from Nanjing University. Thanks for him to give me this good opportunity to doing my PhD, one of my part of my PhD at Weihai. And thanks for the mice that we have sacrificed, and thanks to family and friends. Thank you, and open up the floor for questions. Thank you.
That was fantastic. Thank you very much, Yexuan. And as with all the talks, we are likely to have lots of questions from the audience. And I can see that we've received some questions already, so I'll move straight on to those. So your first question is, how does the SAM system compare to other CRISPR activation systems?
Well, the SAM system was proved more efficient than the very original DCAS9A VP64 only. However, we were unable to compare it with all other CRISPR-A systems side by side. To make our system more efficient in mice, we indeed doing something. We employed the CAG promoter because it has been shown to be amongst the most active polymers 2 promoters across multiple tissues and cell lines.
Okay, so we'll move on to your next question then. So it is, does CRISPR activation work at similar efficiency across all genomic loci?
Well, I think this actually depends on the gene promoter structure and sgRNA designing. I think it showed, like, at least in our experiments, I feel like it showed more efficient in some gene promoter loci than some other genes. Like, I think BCL2 and CD19 works better than some other gene, some other guys like CD4 or something. Yeah.
Thank you. And we'll move on to your next question. How do you design the sgRNAs to get maximum expression?
Well, we could design multiple sgRNAs targeting a broader sequence range of the targeted gene promoter region and look for the most effective guides to use. For example, in some of the sgRNA libraries, we only have like 3 guides or 6 guides for each gene, but actually we are now seeking for the sgRNA libraries, which has like 10 guides for each gene.
That's great. Thank you for that. And I think we've got time for a couple of more questions. So your next question is, have you tested the CRISPR activation in non-hematopoietic tissues?
Yes, we have. I think in the shows that I have shown that we tested it in fibroblasts and memory cells derived from our knocking mice, and it shows that they worked very well. Yeah.
Fabulous. Thank you. And we've got time for one more question. Have you tried to target a genomic locus with more than one sgRNA?
Yes, we are trying a new design as CRISPR-A sgRNA library that I have mentioned, that we have 10 different guides targeting the same loci to achieve highest gene activation. Yes, thank you.
Fabulous. Thank you very much, Yexuan. And unfortunately, that's all the time we have for today. Any questions that we didn't have time to get to will be sent to the speaker after the event. And as with all the other talks in the event, this talk will now be made available to view whenever you want for the next 30 days. That just leaves me to say thanks again, everyone. And I hope you've enjoyed all the talks as much as we have. And we will be back tomorrow at 8 a.m. PDT with some more fantastic talks from our speakers and sponsors. Thank you and goodbye.